All right, so we were talking about the fluid mosaic, and in third hour, this is third hour, so for those of you normally in third hour, or at least registered in third hour, you haven't been in third hour, but registered there, welcome back. Emily, you've often said you wanted to stay in here, so instead of going to, where's the rest of the class? Is it English? You're not in English right now, you are inside. It is a dream come true. This is where we ended up the last time the video was going, and we're using the fluid mosaic model, and you'll see that it is different than the water balloon where you have a balloon on the outside, and if you pop it, everything will come out. And the pattern of the proteins they get put in there depends on what type of cell you have, and some of them they have all the same things, just like businesses have different things inside depending on if they're an insurance place, if they're a store, if they're a construction place. They have different things inside that fit that, but they have some things that are the same. They all have telephone, they all have computer, they all have same things with the cell. They all have ways to communicate with each other, they all have some of the same doorways getting stuff in and out, but some of them are very specific depending on what they are, and so it depends on what the function of that cell is, which proteins get embedded there. And the picture, the polar head, the part that is towards the water, either on the outside of the cell or inside of the cell, is the red part in this. They're not really colored. And the yellow part is the lipid tail, made out of things that we would call fat. It's not exactly like what we would, it would be more like what butter is made out of is that same kind of a molecule, but it's not butter hanging down there, although it kind of looks like it. And then you have the cholesterol, and then these are some of the proteins that we'll go through and talk about. And it is not like a water balloon. You'll see that it's much more flexible than that. All right, now we're to new stuff, so Evan, you gotta keep him on task there. There we go. If you've ever seen the Man from Snowy River, we need to get Evan one of those stock whips, and you can just snap it.
Wow. Okay. That was pretty aggressive, right?
Well, they don't use it to hit anybody. It's just the noise. It's just a noise maker that
Sure there's an app for that. Yeah, I'm pretty sure. Noise maker app.
where you could just take your phone and
Well, Mr. Burgess, okay, we got a bad
So,
app. At the game the other day, they weren't supposed to cheer with their mouths. Like they weren't supposed to make noise, and so they did anyway.
Of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
all right, so they give. They do have that. I don't know if this one's going to actually let me whip it, but we'll see. They give credit to nature for, this takes forever to get to, so we'll just quit. For coming up with this, but even that, how would nature get this put together, make it a double layer, and as you see, as it goes through, there's a lot more to it than just, oh, it's just what keeps the outside, the stuff outside and the stuff inside. It has a lot more functions than that. We're going to talk about some of the proteins. I'm not going to have you write them down, but we'll go through some of them as we go through some of the other organelles. So some of them are channel proteins. One of your vocab words recently, I think, was diffuse. And if something diffuses, what is it? So see, this is now we're cementing your vocab into your mind. Moving how? Apart, spreading out. So if I had something that, I like vinegar, but vinegar's got a fairly strong aroma. If I had a bottle of vinegar here and I dropped it,
I would be.
yeah, it would be bad. But at first you wouldn't even know, but it would come by and all of a sudden Keely would be much more awake and she would be letting me know that that was not really what she had in mind for her environment this morning. And then it would get back to where Kaylee is and it would spread out. It would take a while to get all the way back to where Evan is because it's moving by diffusion, just air particles randomly bumping into the vinegar particles and knocking them around, and eventually they get spread out and spread out as far as they can. And thankfully they would eventually spread out far enough that even though they're still there, we aren't capable of detecting them, so we wouldn't smell them anymore. Channel proteins are just openings that particles will find and will go through just by diffusion. There's more outside the cell than inside, so randomly there will be some going inside. There'll also be some inside going out, but since there's more outside, more will come in just because that's the probability. Eventually it would be equal going both ways and it would just stay the same. Sometimes you want to collect something, so you have to have a transport. It'll actually work kind of like an elevator. It will take energy. Like in the summer when it is hot outside, cooler in your house, and yet you still are taking and putting more heat outside. You're pumping it out. That's what your air conditioner unit on the back of your house is doing, but it takes energy. So if you have parents that are similar to almost everybody else's parents, you get asked at some point, why you don't shut the doors. Are you trying to cool off the whole neighborhood? And I found as a young son that Dad was not looking for the answer, not the whole neighborhood, but at least the backyard, Dad. That didn't go so well. I think I got to clean the garage or something. That was my dad's.
You actually said that.
Oh yeah, yeah. Well, it's Dad. You learn things, you try things out, and Dad's answer to everything was when he was annoyed, I have a job for you. Okay, yeah. Let's go do the job.
Why is it blue but not red?
Because it's, you don't burn it.
And
I
it,
thought blue was an important thing, but it's also black, which means we don't have to write it.
the red is my edition. The blue came with it pre-made by
Okay.
now I forgot her name, Sylvia Mader. She's the one who wrote the textbook, and I don't think she actually did this, but somebody working under the auspices of Sylvia Mader put in the blue just to make it pop. In case you were a little bit tired, it would wake you up, along with a drink of refreshing
Which made me
water.
worried. I'm going back to sleep.
Yeah. All right, so sometime then you'll see, you'll see those. We're going to have to use some of that currency. When we talked about the factory, we needed some money, some currency. The currency that we're going to use is actually little packets of energy. It's going to take some every time we want to move something across the cell. Sometimes cell recognition, and it actually, they make it look like, and they probably, some of them that have it, they get programmed to pick up pathogens. Most of the time, they're going by, if we're talking about Keely, since she's now that blue, 3 words in blue have got her wide awake and with us here. as her cells have stuff going by, and they're just checking everything. Keely, 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 Keely. All of a sudden, not Keely. There's something comes by and they recognize this doesn't belong here, and they can do other. They can call the state troopers, which would be in the bloodstream, the white blood cells, to come and pick up this not Keely thing, or they can force it to exit into the nearest cell, and the security guards inside that cell will take care of it, but they will recognize what is Keely and what is not Keely, and they will treat it accordingly. Nature can be harsh. Receptor proteins are signal molecules. For example, let's say that hopefully it doesn't happen. The first part, hopefully it does. There's some ram, maybe they call them lady rams, hits a spike down the line. Maybe it's cross court, I don't care. And the person in middle back dives for it and makes a fantastic dig. But in doing so, as they laid out and they landed on their knee pads, their knee pad didn't stick right where it's supposed to. It kind of slid down, and you hear that not very satisfying squeal as part of the middle back player's knee ends up as part of the finish on the court there, yes. And you look at it and you get that, it sounds so innocent. Oh, you got a strawberry. And it can be fairly deep that it's rubbed, that floor burn that's on there. The knee cells around that knee cannot fix that very fast. So they send out signals that are picked up by cells all over the body that, hey, these are the proteins that we need made to fix the knee skin cells. And so you have factories all over your body making those extra proteins that get put into packages and shipped, and so your knee would heal pretty quickly if you didn't have to show up and practice the next day and do the same thing or have another game, and you keep going. Most volleyball injuries get recovered during basketball season. It's just the way it goes the next, because you get new injuries in basketball, but the ones that you get in one sport, they heal up in the next one because you keep doing the same thing over and over. But you heal fairly quickly because you have the whole body sending out these signals, and each one of the cells can pick it up. Hey, we're going to keep going. We need what we're going to do, but we're going to up the quota on whatever else this skin cell is, protein, and so it goes around and it will fix that as well. Enzymatic proteins are ones that, as it comes by on the outside, it can actually change things that are going by through the bloodstream. On the outside, it will fit in. It actually changes the shape because that's how proteins get changed. They change what's called the conformation, or just the shape of them, and then they move them on. Sometimes they take them in, sometimes they just move them on, but the outside of it will help carry out the reaction. Sometimes they make these enzymes, which are important because a lot of the chemical reactions that keep us alive really wouldn't happen below boiling point. You need to add enough energy to make them happen, which we aren't all that comfortable around boiling point. 200 degrees Fahrenheit, 100 degrees Celsius. We're not in very good shape long before that. So the enzymes actually make it so that those reactions will happen somewhere around, well, hopefully for this school, somewhere between 97.2 and 97.7, because we're all somewhere in between 97.2 and 97.7. If you are above 97.7, you probably should stay home. Just saying. I've never seen, on any Friday that I've checked, I've never seen anybody above 97.7. Anybody in here ever had it read above 97.7? Without coffee in front of you?
Dude, I got so scared one day. It said 103, and I was like, I'm dying.
I tried to send her home.
I am literally dying right now.
She did, but then she was between 97.2 and 97.7. Yeah. But I'm sure they are fantastically accurate. So the enzymes help all the reactions happen in that narrow space. And the junction proteins hold them together and let them communicate. And it's kind of like plugging computers in a network together. All the cells are connected together. And if you plug one to the next, to the next, to the next, when you get some kind of a signal, let's say that you're out for a walk, for a jog, riding your bike, something, and the neighbor left the back gate out and their angry dog is chasing you, then suddenly you have that fight or flight, depending on the size of the dog, you're going to go. It wouldn't do very well if all of a sudden you decided to run, but it took a while for the signal to go to all of the cells, so one leg was ready to go, the other one was still waiting for the signal to come, and it's nice that the whole body gets the signals all at once and everything goes at that. So as we plug them all together, the communication is pretty much instant throughout the entire body. So here are just pictures of it. We'll go through how they are. The difference, the channel proteins are just open. This one is not. It's going to take some energy to put it. There was in the last animation, they showed one of those, but I wasn't going to stop it. We'll go through. When we do the transport, this is passive transport, doesn't take any energy. This one will be active transport, will take energy. And we have 4 categories, and they're just going to tell you what's coming up. And all of this stuff will be later, so even those of you that feel compelled to, I'm going to write this down. You're going to write it down, and then you'll write it down again. It's up to you, though. You've got the pens and the paper. The first one, nucleus and ribosomes, and they go together because we're looking at the cell as a protein factory. The plans for the protein are in the nucleus, and the ribosomes are the machines that make the protein, so we have to get the plans to the ribosomes, but they work together. Endomembrane system is the transport and packaging system. It will adjust the proteins as they get made, other things as well, but we're going to stick with the protein thing. And then it puts them in packages, slaps a label on where it's supposed to go, and sends it out, and one of your workers is going to take it either to the edge of the cell to put it out in there if it's for some middle back's knee to get repaired, or if it's inside the cell, it'll take it to wherever it's going to go inside the cell. Energy-related organelles, 2 different types. We have, what's our energy? The powerhouse of the cell. You guys, mitochondria. Mitochondrion is one, but she's right, we all have a bunch of them, so mitochondria. Plants have one. What's the one in plants? Sunlight into chloroplast. We got it. The cytoskeleton is going to be the structure of the cell, gives it its shape, and it acts as those, we talked about those catwalks up above where the factory is. They also act as the catwalks for all your employees to get around in there. They don't just randomly swim through the cytoplasm that's in the middle. They have to move along something. They go across a catwalk that's made out of cytoskeleton, which each one of your cells is right now actively building and tearing down and rebuilding somewhere else, depending on what the need is. Just look at me. I don't know what that means. That she's ready. Okay.
Go back.
He wasn't ready.
I was not prepared.
Why is so much of it black, not red?
It's coming. It's coming. I'm just giving you a break.
Okay, thanks.
Oh, let me do that. See if I've got my whip for Keely here yet.
Did you happen to put in the...
I looked at it. I haven't put them in yet.
Okay.
Which one were you? The assessment?
Yeah.
No.
Okay.
Sound 1.
You just have to make the motion. That's one time.
Oh, I do. That's for you, Keely.
Oh wow, that one.
Luckily, it's not on the screen. No students were hurt in the filming of this.
I guess you do have to preface that.
What did it say?
Cytoskeleton. Maybe we should have you and Keely change places. Cytoskeleton. All right. Nucleus communicates with the ribosomes, that's just what I told, to control protein synthesis. And we will go through, that will be the whole part of the first section, which will be the nucleus and the ribosomes. Endomembrane is going to have its own enzymes to make its products, and it's going to put them into packages that we will call transport vesicles. And then you will have little workers that will slowly move along carrying these huge transport vesicles from one end of the cell to the other, going along the cytoskeleton. Haley, are you a setter? I don't think I. That's fantastic. Otherwise, I'd have you hitting right side. You're left-handed. You got a left-handed girl on your team. You got to have them hitting right side.
No, it's right-handed.
You hit right-handed? Oh, then never mind. Go ahead. Go ahead and set. Oh, she hits right-handed too? Do you have any left-handed people that?
Because I'm not left-handed. I can write with both hands.
Ambidextrous. Which do you normally write with? Oh, she's just black and yellow, black and yellow. Hey, yellow and black. Let's mix it up. She's a little
Different.
it's going up.
Oh.
Yeah.
I made the hand motion as I was writing.
All
I
right.
have to go to the corner with that.
Big Bang Whip. It's in HD. If you, I don't know what's high definition about it, but that's just sound one. I don't know if there's more sound.
I can't hit with my left hand to save my life.
Talk it out now. Teen depression symptoms. And I'm whipping. All right. Two types of energy-related cells. Ms. Hoyting can let us know. Mitochondria found in all cells. Chloroplast just in photosynthetic, mostly plants, but there are some protists that have it. But even they, it's not like you have one or the other. All plant cells have to have the mitochondria. They have to have the power plant. They don't run on the sugar that chloroplasts make. We happen to steal that sugar from them or from something that ate them, depending on what. If you're eating pizza today, you are going to get it from the wheat flour if you choose that particular option, or I don't know what gluten-free pizza crust. Is that rice flour or?
You usually don't use rice flour. It's usually like tapioca or
Potato.
Yeah, potato flour. It's usually
Garbanzo beans, but that wouldn't be very good pizza flour.
not really.
Whatever it is, some other plant that has gotten the energy from the sun, and then we get some from the cow that ate the grass that got the energy from the sun. So we're getting it from them. But even the grass itself has to have mitochondria to turn the sugar into energy as well. And then the cytoskeleton is just this network around everything that keeps a shape, and then it is the catwalks. get to your employees, which we're going to call motor proteins. They are going to walk this stuff everywhere, and if you happen to feel tired, maybe it's because you're just feeling for all of those little motor proteins that have to endlessly haul these huge transport vesicles around the cell, and that's what's making you tired. Kaylee, how long do I have today?
13.
I have 21 different whip sounds. No, no. They might have more that I could buy.
That one's
That's a freshie whip.
Oh no.
I should cop a real whip. Practice just. That'd be
Oh,
cool.
that was windy.
that wasn't me.
Golf whip.
Oh. That's why.
Double whip.
Do you remember Jana from Camp Haley?
I like that one.
She just won her golfing tournament. Oh, good. She won her districts for golfing. Jana? Wait, which one was
But Jen is so good. I know Jen.
How do you know her?
That was his husband too.
Yeah. You did? Oh, you went the year before, didn't you? Oh, you got. Wait, which one's
Yeah,
Jana?
you did go to that camp.
Yeah, we went this year.
So you know like Jana, Brenna.
Yep, I know Brenna. She's a cheerleader. She's so cute. Now I know who you're talking about. Yeah. I didn't talk to them.
Those are Tyler's good
That's a
friends.
real whip. All right.
Brenna.
Easy, big fella.
Apparently Brenna's being annoying right now.
Yeah, well Brenna's
All right.
been number one for 2 months now.
You don't have to draw this. You're gonna get a picture of this. I will get you a picture, and we will label the different parts, because you will get to label the parts at some time, as well as we go through all of these organelles. Now this is obviously the drawing. This is a stained. It doesn't actually come that color, but a stained one. This large thing is the nucleus, nucleolus in there. We'll talk about what it does. It has to do with the ribosomes as well. And then here the endoplasmic reticulum, which most of you are fabulous spellers, but in case spelling is not your forte, this will be the way that we spell endoplasmic reticulum, so that most textbooks, after they put it once, they put ER in parentheses. And so if you put that on a test, I will know that it's endoplasmic reticulum. I will hope that you know, if I just write ER on there, that what it is. And it shows it just this purple kind of, what they call them is flattened saccules. They're just like these squashed little channels there, and some of them have, if you're sitting up close, you can see, looks like it has measles or chicken pox on it. That will be the rough ER, the rough endoplasmic reticulum, and some of it's smooth, depending on whether it's making mostly lipids or proteins. But they just show it in one little spot here. It's actually everywhere. So again, if you're, I don't know how far back you can be, on the back is actually even probably better. You can see this part that looks like a fingerprint or thumbprint on there. That's the endoplasmic reticulum throughout the whole thing. It's not just in one little section, but if they actually put it in everywhere, you couldn't see everything else. Just like it here, it's really hard to say what anything else is because it's all covered up by endoplasmic reticulum. We will not be able to see. The best we can do with our microscopes is 400x. This is 10,000x to get this. So that's a scanning electron micrograph. We don't have that capability, unfortunately. But if we did have the money to buy those, I'm sure it would go somewhere else anyway. We would have a nice stadium. And this is animal cell, typical animal cell. This is typical plant cell. Very similar. The membrane is still the yellow in this one going around. Same phospholipid bilayer. But now it has a cell wall that's around it, which is one of the differences. It also has a chloroplast, or a bunch of chloroplasts here. They're seen over on the side. And we will see chloroplasts. That's not a typical arrangement of chloroplasts that we're going to see. You're going to see them as, I don't know if my green isn't working on here. Animal cells are usually rounded. Plant cells look like little bricks. They're square because they have this lattice work in there, and they actually are three-dimensional little bricks. The ones that we're going to look at are going to be very flat. They're squamous is what biologists say. And we're going to look at a leaf, and it's only 2 cells thick. So you can focus and you'll only see either the top cell or bottom cell. That's why every biology teacher uses the same plant for that. And it's easily obtainable from any pet store, except for the first quarter of the year. So I've kind of switched things around because every biology teacher in town is buying all of the, and I forget what, I think they call it Anacharis, and the biology books call it Elodea, but it's the same thing. But they're square, and ours will have, you will see little chloroplasts, and if we're fortunate, we get some fresh stuff. Depends on where it's, how long it's been shipped, how long it's been kept there. You'll actually see them moving, and they can do what's called cytoplasmic streaming, and they'll be moving around inside. After a while, they're pretty stationary. They're just sitting there. The plant still seems to be living, but after a while, they stop moving with that. And the center of it here is, everything is stained so you can see it, is this huge, looks like a lagoon, a reservoir, a water tower. It's just filled with water. Now, we aren't going to be able to see that, but you're going to be able to infer where it is because you'll have all kinds of these chloroplasts everywhere except right in the middle. And where there aren't any chloroplasts, you can infer, well, that's where they are. But now, unfortunately, as this comes over, you can have some room, so they actually could slide over top of it, and then it makes it harder to see where they are. But sometimes you can see, well, there's a big empty spot, there's nothing. That's just where it is, but since we're going to look at it, everything is clear, we'll see right through it. You can't see this big vacuole, central vacuole here that will hold the water. But when you get thirsty, you can either open your, I forget, what's the name of yours?
Mine's Josh.
Oh. Well, I thought it had the name of, like it was human something, or...
No, it's David's.
Oh, healthy human, okay. With that, you can take a drink from your hydro flask or your water bottle, or you could go to the water fountain if it was. They actually are all working, they're just COVID requirements. They don't have everybody using the same one. So you have the big tank outside, the big cooler. We can go get it. Plants don't have that opportunity, so they store up in case there are days, or if you live in Omaha, months without any rain in between. And that's why if you have a plant, I almost don't want to put this on camera, but if you have a plant and you got branches out and leaves and everything is full of water, as they use up the water and it hasn't rained for a while, they start to shrivel, get smaller, and it loses some of the pressure, and the plant starts to wilt. And then you water it again and they fill up, and There you go. If you have a hydrangea plant, the ones that have the great big globe looking, and they're usually either pink or blue flowers, this time of the year is when they really start to bloom and they're beautiful. And if it's very acidic soil, it's blue. If it's more alkaline, you get the pink. You can make them go, you can actually change them. You put some coffee grounds around them, they'll turn more blue. They're a little bit acidic. Those plants are very sensitive to the lack of what's called turgor pressure, and it gets really hot and they'll all wilt. If you water them for just a little bit, they'll pop up, or when it cools off, then they will pop back up again. But a little bit of water and they'll be look real healthy. Otherwise it looks like, oh, I think they're dying. They're not dying, they're just running out. All of these water towers are running out of water. All right. And what did you say, 13?
You have 1 minute.
All right. We will start tomorrow with the nucleus and the ribosome.
Why couldn't I say it if I was one?
Bye, Excel.
Why couldn't I say it if I was
And Rendell and Emily.
Yeah,
You're
let me
watching it, that means you were right side. Yes, you thought you were awake because you're staying right at the board. Oh, wait.
All right, we're going to start the 4 things. We already started talking about the nucleus and the ribosomes. And here are, we talked about, maybe it was the other class, we talked about measles with one of them. You guys probably don't get measles. You have a vaccine, the MMR, mumps, measles, and rubella, that take it. We didn't have them when we were kids. We all got the measles at some point. You didn't want to get the German measles. They were strong, but everybody's got the measles, and you got these red bumps all over. These are actually ribosomes on the outside. Some of them are just free in the cytoplasm, the goop, the jelly-like stuff that's around all of inside of the cell. But some of them, these are ribosomes. These are what actually make the proteins. And they're on part of the endoplasmic reticulum, ER. It even just says ER lumen here. That's just the inside. Even they don't like typing out endoplasmic reticulum. All right, RNA, a nucleic acid, that's what the NA stands for. It is like DNA, except it has a different sugar. Instead of deoxyribose, it has just ribose. So it's ribonucleic acid. And you've probably seen DNA like a twisted ladder. RNA is just 1/2 of that. It's like you took the ladder and cut down right down the middle of the rungs. That's what RNA looks like. So it's half of DNA. And then there's one of the rungs is different than the DNA as well, but we'll get to that later. Messenger, this is the one that we talk about the most, messenger and transfer RNA. Messenger RNA is what takes the code from the DNA out to the ribosome. The DNA is the code, but it can't leave the nucleus. It has to stay in the nucleus, so that code gets copied onto the messenger RNA, which actually is the exact mirror image of it, or the complement of it, because it's made off of the DNA. And then it goes out to the ribosome, and the DNA zips back together and twists up again. And again, hopefully when we talk about how these proteins are made and you see how it's made, you're going to think there is no way that this happened just by chance. This is all kinds of reading and machines, and it looks like computer programming, and it is. And textbook, the secular textbook that I use even compares it to computer programming, which is hard to think that that would happen just by chance. Now the other type, we are going to have another. You're going to have mRNA and tRNA, the transfer, and those we'll talk about. They're going to be really important in the making of the proteins. The rRNA also is, but it really just, it makes up the ribosomes themselves, which will make sense when you see how the ribosomes work, that they have to have the same code that lets these other RNA things fit in there, kind of like little puzzle pieces. But we aren't gonna, I'm not gonna mess with that. You can get all the details in there, and you can't really see the beauty of the whole forest because I'm pointing out too many individual trees for you to see. But the ribosomal RNA is what makes up the two parts of the ribosome, and the ones that you saw on the ER there, those are always together. The ones that are out in the cytoplasm, there are two, and when a code comes, then the two of them come together, and kind of like a clamshell, and then they will make the protein. The ribosomal RNA is actually made in the nucleolus. That's that little, like, looks like the pit or the seed inside of the nucleus. That's what it's made in there. There's a nuclear membrane we saw. Let me go back real quick. Here has all of these pores in it. And this is actually a scanning electron micrograph of it. Looks like a golf ball on here, and you can see all those little dimples. Those are the pores that let things in and out of the nucleus. Lots of stuff going in and out if you need that many doorways, and I'm sure it's pretty much completely covered. There's just part of it here where the micrograph can only focus at one level, so as you went in and out, you would find all of it would be covered with that. So that nuclear membrane, similar to, but it's a little bit different. It's much more, it's not near as flexible as what the cell membrane is. And this one comes with gates. You're going to see that we talk about the cell membrane being a way for stuff to get in and out, but it's completely different. This one actually is a gate, a thing that opens and closes to let things in or out and keep things that aren't supposed to get out from going out, or make sure that the wrong things don't get in. It's a completely different process getting things in and out of the cell. Has to do with that double layer of the cell membrane. Has nuclear pores, and it just lets the passages go in and out. I've already said all of this, so I'm going to take a minute and just think about those Chaldeans and the laws of the Medes and the Persians. And
How do you know that?
All that jazz.
I actually studied when I was in school.
I studied, but I don't even remember.
That was already ancient history even when I was in school. It's not between now and then. That was a long. No, we already had Hammurabi had already made his code, and we had our own rules and stuff. Are you studying about the laws in the Medes and Persians right now?
Yeah.
You can't change them.
Can't change them.
No.
Why not?
It's
Because
that
That's
which
just-
means law.
That's the thing, yeah, it's just.
Yeah, I know
Once
that.
the
I've
still
been
goes
asked.
on.
That was the problem with Daniel once he got put in the lion's den. The king didn't want to put him in there,
Bro,
but
if it's not multiple choice.
But Darius or Darius, or, well, it depends on how different people pronounce it. I do too.
I'm screwed.
He
Wait, so he
extended
didn't want to
the
put him in there
country.
before he was in there?
No, he didn't want to.
Oh, I thought you put him in there and then at night.
No, he didn't.
didn't.
Yeah, he didn't. He was tricked into doing it. That they signed him. They made him. Yeah, so they set him up. Oh no, staple it.
No, I think I did good.
Well, does it say Olivia and Mary on there? Okay, I'm Mary, I guess. I got it. She spells Schmidt different than I do.
Spell her name wrong?
No, I put Mary, I guess. Olivia.
She spells Schmidt.
I still put your name on it, though.
I-G-E-S-S. All right. Did I reviewed enough?
Fonder's law.
All right. Ribosome subunits, one large and one small, are assembled in the cytoplasm and used to make proteins. That's what, when you get to, and you're going to have a wonderful quiz on organelle function, ribosome, protein synthesis. It makes the actual proteins. That's where this all happens. You'll see how it does. It has to get the code from the nucleus, and then it is just the machine, has trucks deliver things to it, and it puts them together.
We made an edible food mold of the cell.
I used lemon.
Jello? Yeah.
I use Rice Krispies.
Use what?
Rice Krispies.
For
The base.
A what?
I used cake. It was basically like lemon cake. Is that good?
And then
was
I
this
had, like,
pre-gluten-free days?
No, it was gluten free too.
Okay.
Ew.
I remember
Maybe
she
if you
would
put it
bring
in the
these
microwave,
really
it would.
good
No, it was good. It was good. My stepmom's really good at making gluten free cakes. You guys are so rude to my.
Diet?
Yes.
That's all right. You'll probably outlive all of us. And then it just is. See?
Nope, I still eat just as many burgers.
All right, we got everybody. Yeah.
No. How dare.
How did?
Change was not of the law of the Medes and Persians. I could undo it. My kids, well, it's
You're
not a
stressing
ring.
now?
My kids do have a tube of wax and a stamp that they used to send to their buddies. They would send letters that they sealed with wax.
Why?
Because it's kind of cool. They like doing things old fashioned.
Weird.
They love to wear the dresses like from the 1920s and 30s, the big frilly things.
They wore dresses?
Oh, my
His
kids,
daughters.
my girls wore dresses, but
Oh, I
just
thought you were talking.
all right, proteins made of these different ribosomes are used in different parts of the cell or different parts of the body, as we saw in dress yesterday. Mary, I guess, was showing off what's left of a strawberry, a floor burn from volleyball, and the proteins used to fix that came from all over, but mostly from that area, but they came from all over her body was chipping in. And proteins from the free ribosomes are used in the cytoplasm. They are just made to order, although some of them get put into the Golgi apparatus as well. But the ones that are on the ER, actually as they get dumped into the ER, as they go through, then other things, it's kind of like the assembly line continues, and it puts other stuff on it and starts to fold it into the right shape. And proteins from the ribosomes attached to the ER are deposited into the ER, into the lumen, the inside of it. This is the part that's supposed to go quickly because it's not red, but I have these overachievers.
I don't know if I'm offended or not.
Okay.
I don't think overachiever is actually any kind of a put down.
Wait, we didn't have to write that?
No. Only stuff with the red. We have to write.
Yeah, we were on
Well, yeah, you're going to be smart. You'll probably end up being a doctor.
Richard, what does this do?
You know who you're talking to?
I don't even know. Oh, that?
Maybe the first
That's
OJ
for
high school.
lacing a baseball glove.
Is it really?
It is.
Huh.
I had to relace my glove for my son to play softball with.
From OCD.
It broke. All right. So here's the whole process, and then you'll actually see it in animation. This is the nucleus, all the pores. Here's one of the pores, that kind of a cross section. The DNA is the blue part that's twisted, and in between there are the rungs. And the rungs are actually, this is the part that makes it recognizable. The rungs are where the language, where all the code is on the rungs. And as it untwists, and there's a special machine that untwists it called helicase, and then from that, this messenger RNA, the green kind of worm-like thing that's coming out here, is made off of the DNA. The DNA has to stay here. This gets actually carried outside. There's something that I'm going to have to get that fly yet. And then when it comes out, it finds there's 2 subunits here, or it could be on one of these that's already on, the subunits are already put together on the ER here. But these 2 come together, and as it slides through, the code off of it gets read, and there's actually trucks called transfer RNA that bring in these, what looks like blue beads here, bring them in one at a time, and the right ones get loaded depending on what the code is on the messenger RNA. And when it's finished, it gets put into the endoplasmic reticulum, and then it gets folded into shape. You have to have it in the right order. That's the first. The primary structure is just having the right amino acid. There's 20 amino acids, and you have to put them in the right order. But that protein won't do anything until it's actually folded and molded into the right shape, and then it can be used in there. If you remember, and I'm asking a lot, I know, but if you remember back to the brain eaters last year, the prions, when the protein got changed, it was exactly the same stuff, but the rogue protein just rearranged it, and all of a sudden it went from doing what it was supposed to do to putting those spongy holes in the brain, so people got CJD, or mad cow disease, bovine spongiform encephalopathy. It's just because that protein just twisted it a little bit, and it went from doing what it was supposed to now it was actually causing a disease. So the shape of it is what makes it either work or not work. Hopefully this will. Be patient for a little while. Patient. Patient. Here we go.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a 2-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins.
These are just trucks that are bringing. This is the protein down here.
Builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported
and it has
to
to
other
have
parts
the
of
right
the cell
code
by
up
molecules
here.
called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function.